Hello and welcome, future scientists, to the first episode of DNA Battles, where we explain a study recently published in Biology. Today we're talking about a study on bovine defenses. This is a very exciting study recently published to the scientific world. It was completed by a team of researchers, so here are their names, and here's the title of the study. So this looks quite daunting, so I'm going to explain the title a little bit. Cows are part of the bovine species, and Staphylococcus chromogenes SC are coagulase negative Staphylococcus CNS that cause bovine subclinical mastitis in cows. Now, this is a painful infection in the udders of cows that impacts the quality and quantity of dairy products. The researchers wanted to unveil what is happening in the udders of cows with SC mastitis. So they investigated the response the cow will have to subclinical mastitis and how this relates to CNS. So CNS are normally found on the udders of cows, but they specifically look at the SC species of CNS because these are known to cause infections such as subclinical mastitis. So how is the cow fighting against this infection, and how does the cow's immune response, which are like the soldiers in this fight, impacting the SCCNS? Before we discuss the results of the study, let's go over some definitions. So, if we imagine DNA as a huge cookbook of information, then we can infer that the researchers started reading this manual to understand how all the different ingredients, the genes, make up the recipes, which are the proteins. And these are actually controlled by the genes. They want to find out what orchestrates gene expression. Additionally, gene regulation controls when and how genes are expressed. Some genes are expressed all the time, while others wait for their cue. They respond to signals from the environment or the body's needs. This study also focuses on epigenetics, which are changes in gene expression that do not alter the DNA sequence, which is the C, G, and AT dinucleotides that we can see on the right. DNA methylation is an epigenetic modification that involves adding a methyl group to the DNA molecule at a CpG site, which is just the CG dinucleotide. The researchers analyzed the epigenetic modifications that regulate the host, so the infected cow's, immune response to SC mastitis. Basically, they look at DNA methylation pattern to observe this. This study employs innovative methodologies such as the introduction of methylation haplotype blocks, MHBs, to more precisely measure DNA methylation patterns. Instead of focusing on individual genes, they investigate MHBs. So these are like whole neighborhoods that have distinct DNA methylation patterns. They want to find out how regulatory effects impact the behavior of genes, improving the specificity of results. The results are truly groundbreaking. Let's imagine the genes in the udders of a cow with SC mastitis that is fighting against CNS on the right using their immune system response, their T cells, on the left. The researchers found that there were enriched pathways and biological processes that were associated with the cow's response to the invasion of CNS into the mammary gland. Basically, there was positive regulation of T cell activities that was associated with the cow's immunity to mastitis. Imagine T cells like the soldiers of the cow's immune system, getting pumped up with methylation, now ready to recognize and fight off the bad guys, the CNS invaders. The researchers found that certain MHBs actually play a role in boosting this immune response. There are hypermethylated gene neighborhoods and co-methylation in CpG sites that enhance the immune system's response to CNS. They influence the expression of genes involved in immune function. The DNA methylation patterns within MHBs will act as a signal and influence the expression of genes responsible for the cow's defense against CNS. Basically, genes in MHBs will undergo changes in expression patterns that allows them to mobilize and eliminate CNS through the amplification of the immune system's response. Welcome to the part of the video where we discuss why this study wasn't just completed for the sake of science. As we know, mastitis is a painful condition for cows, and any people that care about animal welfare and the well-being of dairy cows would be interested in this study as it aims to improve the health and treatment conditions for cows. Additionally, This study has a large economic impact. The dairy industry is worth billions of dollars and plays a very significant role in many economies. Mastitis can lead to decreased milk production, increased treatment costs, and financial losses for all dairy farmers. This research, which helps improve the management of mastitis, will indirectly affect food prices and the livelihood of all of those working in the dairy industry. There's also a large implication of the results of this study on food safety, as dairy products like milk and cheese are staples in diets across the world, and mastitis can affect the quality and safety of these products. This study, which helps understand and prevent mastitis, can lead to safer and higher quality dairy products for consumers, as well as a higher quantity over time. Not only do the results help shape our understanding of how the cow's immune system responds to mastitis infections, but this understanding will help create treatments for bovine mastitis. Treating infections such as mastitis on a farm is difficult, which often leads to the overuse of antibiotics as treatments. However, this has a large negative effect because this leads to widespread antibiotic resistance across farms. These results will help in developing more targeted and effective treatments for mastitis infections, which could reduce the need for antibiotics in dairy farms. As we know, antibiotic resistance is also a global health concern. So these findings and new developments will help us to fight antibiotic resistance, which will impact many organisms, including humans. However, it is important to recognize the challenges faced by any scientific endeavor. In this study, dealing with mixed cell populations was a limitation. This means that milk producing and immune related cells were mixed in the milk samples used. Future research requires more advanced techniques to separate the cells, so the researchers suggested single cell sequencing to refine our understanding and provide a clearer insight into specific cell types and their contributions to the results. This will also help create a clear connection between DNA methylation patterns or changes and specific cell types. Additionally, this study used a small sample size, which restricts the generalizability of the results. This means that in order for these results to be applicable to more populations, these findings need to be tested with a larger sample size. So, more cows in the subgroup with SC mastitis and also in the subgroup without SC mastitis. The larger sample size makes the results more statistically sound and improves the reliability as well. Another limitation is the lack of bacteriological examinations in this study. Bacteriological examinations test to confirm a bacterial infection and help identify what type of bacteria causes that infection. This could have helped link the changes in DNA methylation to the specific bacterial infection. Understanding what specific bacterial infection is present in the sample will help unveil what is occurring when bovine mastitis is caused by Staphylococcus chromogenes. For future studies, bacteriological examinations should be completed to ensure that the results are reliable and can contribute to making effective treatments for ST mastitis. What a fascinating study! Let's go over a recap of what we found out. The researchers looked at bovine mastitis, which is caused by Staphylococcus chromogenes. They seek to identify DNA methylation changes associated with subclinical mastitis and their role in gene expression. What they discovered was that methylation haplotype blocks, which are regions of DNA with distinct methylation patterns, receive the signal to boost the infected cow's immune system response. The results help gain understanding of the molecular basis of mastitis caused by CNS, specifically Staphylococcus chromogenes, and how there are methylation changes in the infected cow that are associated with the presence of CNS. We also looked at the limitations of our study, and along with the bigger sample size, it's clear that there is a need for cutting edge technologies to overcome certain study constraints. And although the results have large implications on the well being of dairy cows, the dairy economy, and reducing antibiotic resistance on farms, it's still important to acknowledge the limitations when looking at the results of any study to ensure we are considering the whole picture. I hope that you enjoyed this exciting study, and be sure to check it out if you have any more questions.